My name is Anisha Haldar, and I'm an undergraduate research assistant in the Lisane Lab at the University of Alabama at Birmingham. Today I will be talking about CoSIA, an R package our lab created to measure and visualize transcriptome diversity across model organisms and their tissues. Disease diagnostics is a fundamental part of research in precision medicine. Particularly in this field, there has been a heavy focus on creating tools and methods to better understand the underlying mechanisms of individual patient variants. In doing so, many researchers have looked to using model organisms to study these patient variants more extensively, thus providing opportunities for discovering new disease mechanisms and possible strategies for treatment. However, one of the fundamental difficulties in using model organisms is in determining which model organism is best suited for studying and modeling the patient variant phenotype. Gene expression is a common metric that scientists use for species selection, but there are underlying challenges to using gene expression to compare across species and tissues. That is why we decided to create the R package CoSIA. We wanted to provide a tool for researchers to calculate and visualize gene expression metrics for specific genes they were interested in and compare those metrics across model organisms they were considering using. The R package uses curated non-disease wild-type RNA sequencing expression data from BG to calculate these gene expression-based metrics for various tissues and model organisms. The two gene expression-based metrics used in the package are Shannon entropy and normalized coefficient of variation. The R package consists of 5 modules. The first 2 modules provide tools to convert gene identifiers across the same or different species. This is important in ensuring that the user has the correct gene identifiers they might need for future analysis. The third and fourth modules look at visualizing gene expression data. The third module looks at visualizing the expression of one species across multiple tissues, while the fourth module looks at visualizing gene expression of various species across one tissue. The last module, which we will dive deeper into, calculates the 2 transcriptome diversity metrics, coefficient of variation and entropy, and provides visualizations for each so that users can explore these metrics for their specific gene set across tissues and model organisms. To best show how the calculations and visualizations for expression-based transcriptome diversity can be used, we conducted a small analysis using a monogenic kidney disease panel from Natera. The 2 transcriptome diversity metrics were explicitly calculated for this gene set and visualized in the 2 figures seen in the results section. The first figure looks at the coefficient of variation for humans and 3 different model organisms, rat, mouse, and zebrafish. The figure looks across tissues that have commonalities to at least one other species in this analysis. The coefficient of variation measures the variability of the gene expression for the genes in the kidney panel across the different tissues. Therefore, when the coefficient of variation is high in a tissue, the gene expression values of this tissue are more variable in its expression. Since the coefficient of variation is a statistical measure of dispersion that uses mean and standard deviation, we can make direct comparisons across tissues and species using these values. The second figure looks at the entropy across humans and 3 model organisms across the 6 tissues they share. Entropy is a measure of uncertainty, the likelihood of something occurring. Entropy tells us whether the genes in this set for this tissue have specific expression, which means that there is low uncertainty and low entropy. We can use the scatter plot to compare entropy across tissues and species. After completing this analysis on the Natera monogenic kidney panel, we are able to identify some similarities and differences in gene expression of various tissues across humans and common model organisms. For example, there are similarities in heart tissues across the 2 metrics among all 4 species. This was identified by looking at the high variability and uncertainty in gene expression across the heart tissue in all 4 species. However, in liver tissue among all 4 species, we saw low uncertainty and high specificity, but differences in the amount of variation between model organisms and humans. I want to note a limitation of this analysis. Since kidney gene expression in zebrafish is not present through ReGi, that tissue was not analyzed in the transcriptome diversity analysis, even though this was a kidney disease panel. Though further analysis needs to be done to understand these findings better, CoSIA allows for preliminary visualizations of these metrics and provides a new way of looking at gene expression across tissues and species. We are currently in the process of submitting our package to an R repository and working on building a Shining app to expand the usability of this tool. In the future, we hope to add additional model organisms to this package, expand the gene expression data we use, and add other metrics to this package.